This video is going over mitosis and cell division for leaving Cert biology, and it's the absolute basics. It's a very fast, quick, simple summary. So start off with what is mitosis? Well, mitosis is a form of nuclear division, which basically means that a nucleus is going to divide to form two identical daughter nuclei, just like in this diagram. The DNA is copied, and a copy of that DNA goes into each new nucleus, which eventually will form two identical separate cells. We think of two categories of cells. There are those somatic cells, which we would consider as normal body cells, so in other words, they're not gametes. And then there are the gametes, those cells capable of fusion like sperm and egg, those sex cells. So to make things easy for yourself, when you're considering nuclear division, you think of somatic cells being associated with mitosis and gamete formation with meiosis, that other form of nuclear division. So what is the function of mitosis? Well, that all depends on whether the organism is unicellular or multicellular. In unicellular organisms, mitosis is for increasing numbers. It's a means of reproduction. It's asexual reproduction. Whereas in multicellular organisms, mitosis is used for growth and repair. To fully understand mitosis, it's important to know something of the cell cycle. So the cell cycle is made up of interphase, then there's mitosis, and then cell division, otherwise known as cytokinesis. Interphase is the longest stage or the longest part of the cell cycle. It's really important to know that. In the later stage of interphase, the cell gets ready for mitosis and cell division. It prepares by producing more mitochondria and more chloroplasts also if it's a plant cell. A lot more ATP is produced and stored, and biomolecules such as proteins are produced. But the most important thing is that the DNA is copied, so it's replicated. So really there are two sets of each chromosome, but they're not yet condensed. They're still in that stringy format, chromatin. So at the end of interphase, exactly what is going on in the cell? Well, firstly, the DNA has duplicated. There are two copies of the DNA. It's still in the format chromatin. So there are extra organelles and more ATP has been produced and stored, but there's also more biomolecules like proteins. But overall, the cell has grown. So after interphase is mitosis, and there are 4 stages to mitosis: prophase, metaphase, Anaphase and Telophase, and it's important that you know each one of these and you can say something about them. But it's also important that you can remember them in the right order. Pippa must always talk. That's my rhyme, but you could make up your own. The whole process of mitosis is splitting up these duplicated chromosomes and ensuring that equal copies go into both new nuclei. So this is a duplicated chromosome. It's made up of 2 sister chromatids joined at the centromere. Prophase is the first stage of mitosis. You can see the nuclear membrane is disappearing. Chromosomes are now visible, they're fully condensed. Spindle fibres have appeared. The nucleolus has disappeared, and there are pairs of centrioles, these microtubules at opposite poles of the cell. Metaphase is the next stage, and it's the easiest to recognise because the chromosomes are arranged across the middle of the cell, and there are spindle fibres from each pole attached to each side of the chromosome, to each sister chromatid at the centromere. Anaphase is next. I always think of A for away because the sister chromatids are separated. They're pulled to opposite poles of the cell by the spindle fibres contracting or shortening. They're now referred to as daughter chromosomes. Telophase is the final stage of mitosis, and you can see that nuclear membranes form around those separated chromosomes, those 2 sets. The spindle fibres disappear, the nucleoli reappear, the chromosomes are less condensed, and effectively you've 2 identical nuclei now formed, and the cytoplasm pinches in readiness for cytokinesis. Cytokinesis is cell division, where those nuclei get split into their own separate cells. So in an animal cell, a cleavage furrow will form. The cytoplasm simply pinches away to form separate cells. Whereas in plant cells, vesicles line up midway between the 2 nuclei, and they contain the materials for new cell walls. They'll eventually fuse to form a cell plate, and this forms 2 separate plant cells. You're often asked, what is cancer? Cancer is a group of disorders where the rate and the number of mitotic divisions is uncontrolled. So some cells can acquire genetic mutations, and these mutations give rise to abnormal cells. And in these cells, mitosis happens too fast and too often. This all gives rise to a mass of cells, a tumour or a lump. Malignant tumours invade other tissues. We associate these with cancer, but benign tumours do not invade other tissues. So what can give rise or cause these genetic mutations? Well, viruses, the chemicals in cigarette smoke and UV radiation are some examples. Some important terms to know. Haploid, denoted by a little lowercase n, means one set of chromosomes in the cell. So gametes would be haploid, so the sperm cell and the egg cell. Diploid, denoted by a 2n, means two sets of chromosomes in the cell, and this is generally associated with somatic cells, your body cells. Finally, it's important that you can compare mitosis and meiosis, those two forms of nuclear division. Mitosis, firstly, maintains chromosomal number, keeps it the same, whereas meiosis halves chromosomal number, for example, diploid to haploid. Mitosis produces 2 daughter nuclei, meiosis produces 4 daughter nuclei. Meiosis produces genetic clones, but meiosis introduces genetic variation. So that was a very quick, basic summary of mitosis and cell division. Now try the exam questions and check the official answers. Use your textbook to write your own revision summaries. The very best of luck.